Hello everyone, I am glad today to be here in African Fungus Day and participate in this interesting events. I am Lobna Hajji from Tunisia, from Regional Center of Agriculture Agricultural Research of Sidi Bouzid in Tunisia, and I will present today this management. Today, with the rising food demand, with this growing global population, the agriculture is pressed to feed a population. In addition, the use of conventional or chemical pesticides causes different risks to soil health, to biodiversity, and also human safety. So we need to protect the planet, and that's why it is required to have these eco-friendly practices that protect the ecological balance for our future generation. And here the beneficial fungi represent a fundamental shift from fighting nature to working with it. How this fungi has this role with fungal biocontrol can reduce this chemical dependency and support the soil microbiome health. In addition, with using this microbial biostimulant or biocontrol, we align with the ecological principles, and in addition, we can provide a healthy and safe food for consumers worldwide. Among the most known fungi that are used as biocontrol or biostimulant, we have the example of trichoderma. This fungi is the multi-talented guardian because it can protect fungal pathogens, nematodes, and also index. And this protection is provided by different multiple mechanisms. In particular, we have the competition mechanism when the fungi outcompetes the harmful pathogens for space and nutrient. With the mode of action of antibiosis, when this fungi produces antifungal metabolites that directly inhibit pathogen growth. Third mechanism is the parasitism, when the pathogen directly attacks and destroys these pathogenic fungal structures. And also finally, the trichoderma induce the resistance, the mechanisms of the plant which is attacked with pathogens. In Tunisia, our olive tree is the most important fruit tree cultivated in the country. And since definition, this culture occupies large place in the Tunisia, as it provides 5% of the total value of final agricultural production. 50% of the total agriculture export also is provided by olive oil, and average of 30% of agri-food exports from olive also, and it assure 20% agriculture employment and 10% of direct and indirect source of livelihood. In the southern Mediterranean, Tunisia is the most important olive growing country, and olive is a pillar of Tunisia agro-ecosystem, giving it a great economic, social, and cultural importance. And that's why we will talk about olive tree management with these beneficial fungi. In Tunisia also we had an olive germplasm, and when we have more than 200 varieties of olive that are conserved in the National Gene Bank of Tunisia. Despite this great importance in economy of Tunisia, this production is faced with different challenges. Among them, this modern and mechanized agriculture. That's why the olive grower actually they need to provide high productivity and quality with. In addition, this variety must be suitable for with this modern cultivation system. Furthermore, the olive is faced this climate change impact, which is associated with abiotic stress like salinity, drought, and also some biotic stress like pests, such as Bactocera oleiae and Praise oleiae also. Some diseases like Verticillium dyliae, which is the disease that we will talk about in our work in our laboratory. And also we have a new emergency for worldwide, which is Xylella fastidiosa, which is the olive quick decline syndrome. And this syndrome it's unstoppable, and until now we don't have suitable treatment, and also we don't have a resistant variety to this new disease. With Verticillium dyliae, this disease, as you see here in this photo, it starts with this yellowing of some leaves, and then with wilting, and after that we can have like the death of these mature trees. And the main symptoms that we can know this disease, the main symptoms is this darkening of xylem tissue. That's why we call it them vascular disease. The main management methods that we have, like prophylactic or preventive treatment. We don't have chemical treatment to this Verticillium dyliae. Among these practices that we can have it in the field with the farmers is like to burning, and replacing the affected olive tree by resistant variety. And also we need to avoid this crushing the remain of infected branches. Also avoid handling contaminated soil by the fungus. In our project CIRAM, which is international project under the program of PRIMA, the Mediterranean program PRIMA for research and development, which the consortium is composed by different partners from Egypt, Morocco, France, Portugal, Spain, Greece, Italy, and Tunisia, coordinated by myself and at the Regional Center for Agricultural Research. The main objective of this project is development of tailored approach to address climate change, the desertification, the pollution, and the low-income issue under different smallholder farming system in the Mediterranean area. And we use it here a lab to farm approach that starts from practices of regenerative agriculture, then including them the screening of existing crop variety, microbial strain, and here we will use our trichoderma associated with other microorganisms and also associated with some organic waste biomass, in order to have like a holistic approach or the best strategy for Mediterranean scenario under pedoclimatic condition for us for our Tunisian condition. In our Tunisian case study, and with this approach from lab to field, we are starting from the field and we isolate, we prospect and sample soil and some leaves and some tissue of olive, and then we come to the lab, we screen these beneficial microorganisms, fungi and bacteria, for their PGP traits and also for their biocontrol activity. Then we use under controlled condition these microorganisms to test them if they can enhance or not the plant resilience toward biotic stress and abiotic also stress. And then we do like scale up to validate this lab results under field condition, and here we evaluate the impact of this microbial consortia associated with other regenerative approach. Starting from prospecting and soil sampling, we did it more than 20 farms of olive tree in the region of the center of Tunisia. We choose it Sidi Bouzid and Kairouan. They are the most growing olive tree, and we did it like survey to collect the data about all the usage, all the used practices, and those also we sample soil in the laboratory. We isolate the fungi from PDA, the bacteria from different different media, like LB, Lurea bortani media. This is for the Pseudomonas. ESP2, which is for actinomycetes, nutrient agar for general bacteria. Then we incubated the fungi and also the bacteria, and then after screening these microorganisms, first we did the pathogenicity test, and then we passed to the PGP traits and also the biocontrol capacity. In addition, the sampled soil is characterized with different physicochemical property, like the carbon nitrogen ratio, the electrical conductivity, the organic matter, the granulometry, the texture. Then we correlate this microbial community, fungal and bacterial community, with this physicochemical property of the soil. This Pearson correlation coefficient showed that this the fungal and bacterial community are strongly correlated with soil, organic matter, organic carbon, nitrogen, and water content. And also the relative abundance analysis revealed that the genus of Aspergillus, Cladosporium, Alternaria, Fusarium, Penicillium, Trichoderma, and Verticillium were the most found or abundant fungal genera found in our soil samples. Regarding here, some bacteria which are screened for their extracellular enzymatic activity, like amylase, pectinase, catalase, and glucanase. And in addition, for PGP traits, we screened even the bacteria or the fungi for the hydrogen cyanide, the nitrogen fixation, the phosphate solubilization, and the indole acetic acid. Then also we screened these microorganisms for their biocontrol potential by confrontation methods, and this is direct methods. And then we pass to detached leaf assay when we screen here, for example, some bacteria which are, for example, here we have a B101 with the most inhibiting the development of wilting caused by Verticillium dilae. And after that we have like a microbial collection which will be useful for our further analysis. Here from this collection we choose it, for example, Trichoderma longibracketum and Pseudomonas yamaranum against Verticillium dilae. And we applied three under laboratory condition. We applied here, for example, for Trichoderma and also for Pseudomonas, the direct confrontation methods and indirect confrontation methods and culture filtrate. For indirect confrontation method in culture filtrate, we can have like how, for example, these microorganisms can produce some VOC compounds or some metabolites that are the most active for this biocontrol potential. For direct confrontation methods, these two screen if Trichoderma or Pseudomonas can have this direct effect or parasitism effect, for example, with Verticillium dilae. After that, we pass to leaf assay when we infect these leaves with Verticillium dilae and we treat them before with Trichoderma and also Pseudomonas, and then after 21 days of post-infection we evaluate the disease severity. After that, we pass the third experiment. It is a pot experiment, and here we want to see if the age of we screened 3 age of Shimleli, and after 3 months we determine several parameters like soil biological and physicochemical parameters, some agronomic ones, physiological and biochemical parameters, and also the disease incidence. And here we want to see if this our treatment by Pseudomonas or by Trichoderma, or this association, for example, or associated with other practices, they are dependent or not of the age of the olive tree. We choose Chimleli because it is the most growing variety here in Tunisia. It's local variety. For the first, we find, for example, independently with the methods that we use it, that the two microorganisms have the Pseudomonas or the Trichoderma have an inhibitory effect against Verticillium dilae. In addition, here with the pot experiment and leaf assay also, we found that this combination of microbial consortia with composts could be used to protect and improve the plants from the pathogenic fungi Verticillium dilae. And pot experiments also it can improve soil quality. Among our dissemination impact of this work, we published this work in Journal of Plant Disease and Protection this year, at the beginning of this year, with the paper titled Harnessing Microbial Consortium and Organic Soil Amendment to Control Verticillium Wilt caused by Verticillium dilae in olive plants. Then another work also in olive plant, when we screen or evaluate the olive plant resilience to biotic stress through this microbial consortia with or not with compost. And here we will do it under pot experiment, and we will screen not to for this experiment we will screen different variety. We have it the first experiment, we have it a good experiment with Shimleli, which is the local variety. Here we want to see if this effect is confirmed or not with other variety local like Chetui, and two introduced one, which is Arbequina and Arbuzana. And here we treat the plants under controlled condition. This is pot experiment with composts, microbial consortia alone or associated with composts. We have with compost, and we infect them with Verticillium dilae. And after 3 months of post-infection, in addition, we did it. We determined the physiological parameters like the photosynthesis and chlorophyll, some agronomic parameters, and some biochemical parameters like the stress response by measuring the airways enzyme like peroxidase, catalase, and some other enzymes. MDA, lipid peroxidation, proline, proteins, antioxidant activity, phenols, and others. We determined the disease severity, and also we determined the soil physicochemical month. After 3 months, this is association of the compost and microbial consortia was the best for the whole variety to reduce this disease severity of Verticillium dialiae. Then we want to scale up or validate these results under field condition. And here we have it in our experimental farm in Regional Center of Agricultural Research, which contains three varieties of olive tree like Chetui, Arbusana, and Arbequina. And here we want to see in natural condition the impact of this microbial consortia composed by Trichoderma and Pseudomonas associated or not with compost, and we will see after 3 months the impact of this treatment. And also it was confirmed that this compost associated with this microbial biostimulant was the best to improve, for example, the proteins, the phenols, and to reduce the MDA, lipid peroxidation, which is stress markers. And among our dissemination impact of this work, we published it in Journal of Science, Soil Science and Plant Nutrition in Springer, with the paper titled Sustainable Integrated Management of Olive Verticillium Wilt using Microbial Consortia and Compost Mill Waste. The main achievement of this work and some concluding remark that we have it, that's the combined treatment, this combined treatment of microbial microorganisms and the compost improves the plant vigor, antioxidant defense, phenols, proteins, soil quality, physicochemical quality, and also the microbiological quality. For all variety, the combination of microbial consortium and compost can be potentially used as integrated management for Verticillium wilt. This study is interesting for improving olive tree health and productivity and can give new insights and significant consequences in sustainable agriculture in global, not only in Tunisia. Until now, we have it like supervised two research masters, two profession one. We have it one PhD students working in olive tree, some communication, and also two research paper and two workshop. And we actually funded with new projects, which is EcoFerm Lab, like Living Lab, which will be like a show of our results. And here we will transfer these results that we have it from our work to use microbial consortia as an integrated approach. And here we will boost this green transition to agroecological transition. Our future goals are to evaluate the long-term environmental impact as assessment of this treatment. Even there are biological treatments, we need to have this monitoring in long-term effect. We need to develop a tailored formulation for this specific condition, and also we scale this for our other experimental plots. We want also to explore the use of remote sensing on soil test kits and also ensure the widespread adoption of these sustainable practices that like to transfer the knowledge, capacity building, and support policy. Thank you for your attention, and here my contact if you need any further question, and thank you very much for your attention.